Okay, thank you, Teresa, for the introduction. Yeah, I'm Bess Hardwick. I'm one of the coordinators, and I'm going to start by telling you some basics about LIFEPLAN. It's a global terrestrial monitoring program using a bunch of methods, including these malaise traps that you see here deployed at eLTER stations around Europe. Then I'm going to go into the role of eLTER in LIFEPLAN. I'll tell you a little bit about the current status of the project, and then just a quick word about what happens or doesn't after LIFEPLAN. So we are a global terrestrial biodiversity monitoring project that started in 2020, and we've just now started our final year. So we'll end in end of March 2026. And we have 3 levels of monitoring. So we have the global design, where a bunch of eLTER stations are included. And in the global design, the sites alternate between a natural and an urban location yearly. Then we have the national level, which is in Madagascar and the Nordic countries. A bunch of eLTER stations also in the Nordic countries, and these sites stay in the same location every year. And then we have an even denser network of hierarchical sites that we run ourselves. At each of these sites, we have this 3-point transect, where in the middle we have a malaise trap that collects arthropods, a cyclone sampler that collects fungal spores from the air, and we do soil sampling, where we also look for fungi in the soil. And then we have camera traps and audio recorders. And then we also have these 2 other points along the transects. We have just camera and audio and soil sampling. And these 3 physical sampling methods, the malaise and the cyclone and the soil, we identify the species by molecular methods. So we extract DNA and sequence it and identify the species by barcoding. And from the camera and audio data, we identify species by machine learning methods. Here's our global sampling map, and you can see the eLTER sites here in green. So you can see that they are a really substantial proportion of our European sites. And Europe is a sort of higher density area for us, where we sample at a higher density and get more information. And of about 88 total teams that we have around the world, 25 are eLTER teams. So it is a good chunk. Compared to the other sites, which are various kinds of independent researchers, research groups, universities, all kinds of people, eLTER sites in general have been some of the first that started sampling, and they have performed very consistent sampling. So they've been very nice in that sense. This is partly because the eLTER sites tend to have the best infrastructure and local conditions. So they'll have staff that's employed there year round and electricity and things like that. That's not always a given. After we get your samples and your data, we do quite a lot of stuff to it. So for the physical samples, there's all kinds of cleaning steps, and there's DNA sequencing. And after DNA sequencing, we perform bioinformatic analysis, and then we get to species lists. And for the physical samples, we are currently getting species lists on all of these. For the camera and audio data, we have to collect a lot of annotation data, which is the data that we use to train the machine learning algorithms. So we just tell it what the species look like or sound like. And we are for the audio, we are for some places able to produce species lists now. And for the camera data, we're getting there. How is sampling going? So in this image, I have just the eLTER sites. On the y-axis, they're sorted by latitude here. So the northernmost site is up top. And this shows 4 years of sampling. So these kind of chunks along the time, the x-axis, you see years 2021, 2, 3, 4, 2025 sampling is just getting started now. And I've color coded the sampling week. So each of these little lines is a sampling week by what the status of the sample is. So these are just the malaise samples now on this slide. So you can see that some sites still haven't actually managed to send in their samples. So that's in a bit of an urgent thing to get done. But most of them have sent samples. The blue ones are still waiting to be sequenced at the lab. So you can see that the last couple of years, some of it is still in the queue. Then there's very little pink, which means that those are sequenced but not yet bioinformatics. And most of it is actually in this lovely, beautiful wine color, which means that we do have some initial bioinformatics done, so species lists. Cyclone samples also have still some outstanding samples in black, teams that haven't sent them in. But here we have much more samples that have been sequenced that don't yet have bioinformatics, these pink samples. So that means that we had a very long backlog at the sequencing lab of getting samples sequenced, but we're now starting to get a lot more sequence data. But we've just started running the bioinformatics pipeline for fungi. So we don't have many species lists yet, but we are going to start producing them much faster now than we are getting sequence data. Soil samples is much more sparse. We only do it a few times a year. Again, some teams have samples that are still outstanding that have been sent in. And for soil, the bioinformatics pipeline is the same as for cyclones. So only a few sites have species lists so far. But again, this is going to start going much faster. For camera trapping, so we just have black and green. So black is data weeks that have not been uploaded to us yet, and the green ones have been uploaded to us. Again, a couple of sites there that haven't uploaded data, so that's something that will be very important. But mostly other sites have done a really nice job of keeping their data up to date with us. And the same basically for audio data. And this is just the eLTER site. So when I look at these kind of pictures for the full LIFEPLAN teams, eLTER sites are doing, in general, very, very nicely on this. So what data are available right now? The Malays 22 teams, that basically everybody who sent in samples could have raw sequence data if you want. Raw sequence data is very, very large files, and they're only useful to you if you can run bioinformatics for yourself, but you can request it if you want. But we do also have initial species lists available upon request. For cyclone samples, it's at the moment mostly only raw sequence data. Again, very large files, but if you can run bioinformatics, you can request those. And the same thing for soil. So how do you know if you have some kind of data available yet? Every month we send teams an updated version of this kind of report. So you can see the alternation between natural and urban. 2021 they were natural, and 22 they were in urban, and they go back natural and urban. Each of these little stripes is a sampling week, and then we have all the different methods: soil, malaise, cyclone, camera, and audio. So you can look at this each month and see, okay, we have lots of samples in pink. Maybe I don't want sequence data for cyclone, but maybe I want sequence data for malaise, so I can ask for that. And then you could also see if maybe you haven't sent in your samples. So the teams get this kind of updated report every month. Another great thing that the other teams have done for us is this annotation data. So we've been collecting audio annotation data on our online portal. So we have a lot of this data for Europe and eLTER sites where they've listened to bird sounds and identified species. So that we very much appreciate. And because of that, we're very close to having European models that we could run audio, identify the vocalizing birds in the audio data. So what happens next? We're currently trying to run the result reports to get species lists to teams. We're working on that this spring. Sampling in LIFEPLAN is going to continue until October of this year. We're going to stop a little early so that we have time to process your samples before the full project ends. Right now, you should be making sure that your samples are sent in, sending them in, uploading your data, getting everything on so that you can be included in the data papers and the ecological papers that we are going to write with all the co-authors from all the teams. After LIFEPLAN, I know eLTER has some plans for using LIFEPLAN, which is a method, in the future as well. We are going to try to make the tools for turning raw digital data, sequence data, images and audio into species identifications publicly available, so that you would, after LIFEPLAN, you'd be able to use those for free to run bioinformatics on sequence data or identify images or audio. So those would be available. The sequencing and the otherwise handling of physical samples would then be, after LIFEPLAN, up to the sites themselves. So we won't be here to handle the sequencing and indeed pay for it. But the equipment will remain with the teams in whatever condition it's in. So you'd still have the traps to keep after. So we do hope that as many teams as possible continue using the LIFEPLAN method. These long-term data sets are unusual and valuable, and as a standardized method, it's really great if eLTER can continue with it in some way. Okay, that's it for me. And I think Thomas was going to go next.
Yeah, first of all, thank you, Bess, for a nice presentation. As Paulina already mentioned, if questions come up to Bess, either please post them in the chat, or we can answer a few right now. But if nothing urgent, if no burning questions come up right now, I think we can move on to our next speaker.
I'm guessing it's Otso.
Yeah, exactly. That's what I was going to say. Wrong order. So welcome, Otso. A little bit about him. He's a professor of mathematical and statistical ecology at the University of, oh God, Jyväskylä. Kind of. Sorry, I'm really bad with Finnish names. After finishing his PhD in 98 in mathematics, he realized that he's more interested in the natural world than in engineering, and decided to pursue a career in ecological modeling, which I find cool. And his main focus is in developing AI and bioinformatic methods for classifying species from DNA, image, and audio, and in developing joint species distribution models for making more out of biodiversity data. Welcome, Otso, and whenever you're ready.
Okay, thanks a lot, Teresa, for this very kind introduction. So let me share my screen. Now I hope you see my slides in a nice way.
Yes.
Yes.
Thank you.
So I'm not going to talk about the ecological results or questions. Thomas will address those. I will talk about the state of the methods development in LIFEPLAN. And Bess already presented all the data types and at what stage we are with them. Now, what are the steps we need to take to address ecological questions with these samples? If we simplify a lot, we need to do 2 steps. First one is this classification, how to classify or identify all the birds and bats from the audio, mammals, and some other animals from the images, fungi and arthropods from the DNA meta-barcoding data, how to get these species lists that Bess was talking about. So that is kind of the step 1. But then that's of course not sufficient. Step 2 is then to learn something about those data, to do this inference and prediction. And I realized that I have the same text accidentally also in box 2, but here what I wanted to say is that we need the statistical methods to make this inference and prediction and learn from those data. And I will talk about these 2 steps. So let's start with the species classification and let's start with the DNA. How do we do this taxonomic classification of DNA meta-barcoding? And what we, especially Brendan Forneo and other researchers from LIFEPLAN have been doing is to develop this Optimotu method and Optimotu pipeline. So what this method does is taxonomically aware OTU clustering with kind of optimized thresholds. And it includes this full bioinformatics workflow for meta-barcoding data. Starts from the raw sequences and then results in the kind of data tables that I show here. So this is a very, very, very small sample of the LIFEPLAN malaise sampling data. In total, we have in the global data almost like 1 million species level OTUs in these 8,000 samples that were available when doing this analysis. So the OTU table is this typical table of which OTUs were detected in which sample. But then what is a little bit more special with this specific Optimo2 pipeline is that it yields this kind of a full taxonomy for all of those species level OTUs. So here you see the first couple of species out of these million species that we have in this data. And of course, we cannot always go to the species level. That is very clear with arthropods and with fungi. But sometimes we can with at least 90% probability confidence that we have used here as a threshold. But in those cases that we cannot do that, we call the species, we still give it an ID, a pseudospecies, and then we classify the genus. If we cannot do that either, then we give that an ID, pseudogenus, and in this way we construct like a full taxonomical tree for all the OTUs. And this is very important for the downstream analysis because that helps us to place the species in a phylogeny or taxonomy even if we don't know their IDs always to the species level. So this is the output of this Optimo2 pipeline, which is now kind of ready. There's an archive paper, so you can download this stuff and run it, but a bit later it will be more user-friendly. So now I would say that the bioinformatics methods, they are basically ready. We are happy with them, small tailoring still, but they are up and running. Then if we talk about the audio data, it's mainly targeted to birds and bats. I mean, we want to do some things with insects and amphibians, but birds and bats is really the main target. And as Bess already mentioned, an important part of this work has been this development of this annotation portal, BSG, Bird Sound Global, where we have uploaded a good amount of LIFEPLAN data, samples of the data, these kind of 10-second segments that then bird experts, including many from the eLTER sites, have annotated. And now what is important here is that we produce these kind of strong labels, not only which species are present in these 10-second segments, but also the bounding boxes in this time frequency space of the spectrogram, like where exactly each species vocalizes. So this is called like a strong label in the annotation world, and they really help to improve the AI models. So for birds, we use the BirdNet as a baseline, but then with this kind of data, we can fine-tune BirdNet so that we get much more reliable classifications. So we have had methods development on how to do this. And at this moment, bird models for Finland and Madagascar are much better than the baseline BirdNet because we have worked most on those. But models for other LIFEPLAN sites are in preparation, and especially for Europe, we have sufficient annotation data that we can do like the European bird model. And all of these will be published open source, and we will also publish kind of a desktop versions that download this, so you can run your own audio through your own computer if you like. Our European bat model is under development. It hasn't been published yet, but we have a model that works better than any of the commercial classifiers for European bats, and this will also be published in open source. Then about animal classification from images. So we have developed the workflow, especially by Tommi Mononen, and parametricized that for some of the sites. So here is an overview of the workflow. I will not go into more detail here to save time, but just show you an example result that once we run this pipeline, then through the data here as one example from one site, we will get this kind of a plots, which in this case show like when these squirrels were active and when we could detect the squirrels from this site as a function of day of the year and time of the day. So each small pixel here is a 10-minute observation slot, and then the color shows how much squirrel activity did we pick up. And you see that the data are quite nice and clean in the sense that it follows like the patterns of the sunrise and sunset as expected. And this is kind of test data. So this is here we show how well the classification works for cameras that were not used in the training, so it's kind of independent test data, and the results of these tests are that it works pretty well. But as said, we don't yet have like the animal models for all the sites. We have the methods, and we have shown with a couple of sites that they work. Now we just need to do the work for all the sites to do this throughout the data. All right, that was a brief overview of the step 1, like how species classification. So then step 2, how to then learn from this data. The main statistical method that we are using is joint species distribution modeling through this HMSC, which is hierarchical modeling of species communities, which is a method that we have developed in our group in over the past 15 years, so that we have like a long-term methods development in this area. So this method takes as input, of course, the species data, and not only for a single species, but for all the species simultaneously, because it's a joint species distribution model. And then the predictors, in this example, land cover and climatic data. And then also the traits and phylogenies or taxonomies of the species. They are an important part for borrowing information between the species. And now this is the reason why it's so important that we have this full taxonomic tree also for all the fungal and arthropod data, even if there are a lot of missing species, because then we can utilize more effectively this borrowing of information between related species. Okay, after fitting the joint species distribution model to all of this data, we can do like inference and prediction, like learn how the species respond to predictors, such as climatic predictors or land use predictors or urban versus natural, and then of course make some predictions for the future if we like. And this is not from LIFEPLAN data, it is like this pre-LIFEPLAN project that some of you participated, this global spore sampling project, where we used cyclone sampling, the same method as in LIFEPLAN, but before LIFEPLAN. I'm showing this because we don't yet have published results for LIFEPLAN, but for this we have a result where we applied HMSC to those data to learn about the spatial and seasonal dynamics of fungi, and to bring also some evolutionary insights from the analysis. So this is kind of a proof of concept that HMSC can be applied to these kind of global data, which we will do with LIFEPLAN. But then there is this methodological challenge that how to scale up these joint species distribution models for a million species, which is what we have with Malaise trapping especially. And if I start a bit from the history, when we first developed the first versions of this JSDM, the upper limit was like 20 species, that was the most that we could do computationally. And then we have been scaling up to 100 and then to 500. And then that happened like up to 5 years ago. But then in the recent years, we have really put like efforts on how to do much bigger data with JSDMs, very much motivated by LIFEPLAN. So we published this paper in PLOS Computational Biology, where we simply utilize like bigger computers, like GPU-based computation in servers, so that we can scale up the computations to thousands of species. We can fit HMSC now 1,000 times faster than without these computational developments. But even those methods, they don't scale up for millions of species. So now very recently, this is still a preprint, unpublished paper, we developed this common to rare transfer learning approach with which we can actually fit this model for a million species, literally. So I just want to illustrate here how that works. This is a case study of the Malagasy Malaise sampling with 250,000 species. What we did was to fit a backbone model for the 1,000 most common species. This is what we can do computationally quite easily. And then we borrow information from this backbone model through the relatedness or through the estimated taxonomy for all of those rare species. And now we can fit the rare species then independently of each other in a fully kind of a parallel manner. And what you see from this AUK comparison is that this actually works. we get like much better predictions for independent validation data for all of those super rare species that occur even, once or twice in the whole data by borrowing information in a taxonomically informed way from the common species. And now this is computationally highly scalable. We can do this for million species if we like. So this is my last slide. I would say that at this moment, most of the data that we plan to sample in LIFEPLAN are available. As Bess said, they are not like all in the end of the pipeline, but available at some level. And also most of the methods that we plan to develop in LIFEPLAN, they are more or less ready. So now the priority for the final year of LIFEPLAN is really to address the scientific questions that motivated LIFEPLAN by applying these new methods to the new data. And what are these questions? Thomas will tell more about that. Thank you.
Thank you.
And also there is one question already in the chat, and that is for the bioacoustic data, will the improved annotation models at some point be made available in tools like BirdNet or maybe in your own software?
Yeah, so I think there are two parts of this question that will the annotations be made available, and then will the models that are based on those annotations be available? And the plan is to do both. And as I mentioned already, for the models, we are planning to publish them, I mean, not only as open source somewhere in GitHub that, more advanced users can take them, but also make more user-friendly versions. So we plan to make like in collaboration with Luomus, the Natural History Museum in Helsinki. We plan to make like a web application where you can throw in like a very small sample of data to test how our methods work and get the classifications through this web app. But then also a desktop version that you can download this classification model to your computer for birds, for bats, and then run through any amount of data that you have there. But then also these annotations, we plan to make publicly available so that then also, other methods developers can make better models. And this we are discussing with Xenocanto and other actors in the field, like how to make that in the best way so that, they are most compatible with what everybody else is developing and using.
Yeah, I think Thomas's question has been answered. We have another one. How good is annotation mode for bats, and how does the method compare to BirdNet's 3S clips?
Yeah, so this is more fresh, and it's still in preparation. We are preparing a manuscript, but we haven't yet fully prepared it, or even. And here the annotations are not done by like LIFEPLAN partners like eLTER, for example, but we have one specific bat researcher, Kata Meramo, who has done the annotations herself. So she has collected this kind of bat recordings from European collaborators. I mean, recordings which have been targeted to some species, that there is a label of the name of the bat species at the recording level. But then she has used our annotation portal, modified a little bit for bats, to draw these boxes of where especially these species are vocalizing. And then we have fitted a machine learning model based on those data. How good is the model? I mean, we have compared it to all of the commercially available software. I think Kaleidoscope was the best out of those other software that we compared. And maybe our model is like, I don't know, difficult to say, but 10% better, 20% better, a little bit better than the Kaleidoscope model. I mean, not maybe hugely better, but it's already a little bit better. And we are convinced that we can make it still quite much better by simply adding more annotations and improving a bit the model. And then one major difference is that we will publish this model as open source so that researchers can use it for free and provide this software. I'm sure it's a bit worse than for birds, simply because for birds there is so much annotations available that it's easier to make a good model for birds. But it's reasonable. It does most species quite nicely.
Okay, thank you for answering these burning questions. Then I think we're moving on to Thomas's presentation. And Thomas is a professor of insect ecology at the Swedish University of Agricultural Sciences, SLU, in Uppsala, and a professor of terrestrial ecosystem ecology at the University of Helsinki in Finland. And he is passionate about two things, biodiversity, along with the factors that shape it, as well as distributed science projects. And together with Otso, they are two of the three PIs of the LIFEPLAN project. And Bess is one of their two outstanding coordinators, in case I haven't mentioned it before. Okay, welcome, Thomas.
Thank you. And you can hear me? Yes? Great. So I will talk a little bit about what comes out of the full project and also emphasizing just how new the results are. And in doing so, I will try to address questions which I think are particularly relevant for eLTER. So if I manage to change the slide, then what I believe eLTER is all about is trying to detect changes and trends in our environment and trends and changes in populations and in biodiversity, like envisaged here. And the important thing to realize is that this used to seem like a simple task, but it has got very much more complicated over the last few decades, because what we have realized is that of the populations and the species and the biodiversity that we're trying to detect changes in, we've only scratched the surface. So here for animals and plants and protozoa and fungi and algae and what have we, the black parts are the fractions that we think we know, and the white parts are the parts that we can be fairly certain are still missing. And now what we are trying to do is to detect changes in overall biodiversity based on a slice of that black part, so thin that it's barely visible. Like 80% of our data are still based on birds, and those birds account for about 1 per mil of overall species richness. So the main part is simply unknown, and it's quite evident that to get a good grip on what is happening, what is changing, and what is driving these changes, we really need standardized data on a whole new set of taxa. We need comprehensive data, taxonomically comprehensive data, and that means that we need to start dealing with taxa, which we know from beforehand that they are mostly unnamed, they are undescribed, and they're even uncircumscribed. So it's hard to delimit the units that we're trying to monitor or understand. And that's why that's what LIFEPLAN is about. I mean, Bess and Otso already talked about the idea that we need standardized data on all taxa. That is why we have designed these different sampling approaches to target the groups where we know most of the both known and most of all unknown diversity is. But then the second part that Otso was talking about is that we need a new type of methods for dealing with this kind of unknown or dark diversity. And just why that is important and just how LIFEPLAN is contributing with new results, I will try to illustrate by talking more about something that Otso already talked about, because this is really an important thing. Because what I was just saying is that if we have a set of images or sounds, if we had more sounds beyond birds, or in this case DNA sequences, then we know there's a lot of them, and we know that they differ from each other. So here every blob is a sequence, and it differs from the others by the distance that you can see between the different points in this graph. And now the challenge becomes that we know from beforehand that when we try to put a name on these, based on comparing it to known species, we can only put a name on a few of them. So here there are some who get the name whatever, Aphodius fossor or just blue. Here there's a red one and there's a brown one and there are some green ones. These get assigned a name, but for the other ones, we can't match them to anything known before because they have never been described before, and we can't decide where to cut the differences, the limits between potential species or higher level taxa in this graph because there are no obvious criteria to do so. And this is the kind of starting point that we are dealing with in the real world. And then the question is, how can you get smart about this so that you can still get to a list of species-like entities across your samples, sites, times, etc., the OTU tables that Otto was referring to. And the result, the approach here that we are using is simply, simply to learn from how large differences are between previously known taxa at any given level, starting from a class, order, phylum, family, genus, and species, and then use these known differences between entities which we already know to decide how close variants of the sequence we can group into the same unit. So that sounded very abstract, but what I will show here is that if we are still with the same sequences are the blobs in here, we can only name the ones with a color. The color corresponds to the different names. And here, based on some magic, which are exactly the same steps that I'm now going to describe downwards from here, we have decided that there's probably one family and 3 genera in this different in this set of sequences. And now we need to decide, so what do we do about all the unknown ones? Whom should we group them up with? Whom should we cluster them with? And then what we do is that within each taxon, we learn from the already described material how large the differences between the different units are, because that should be the range over which we can cluster these things into the same groups, into the same clusters. So you can see that within the green genus here, I have drawn, or actually Brendan has drawn, wider radii across the species, because here we know there is more variation, and in this one and this one, we know that there is less variation within the species within these groups, so the radii over which we should join them or cluster them together are smaller. So in the next step, what I do is that I group together units which are close enough to the named ones that they should basically belong to the same cluster, and I give them the same name. Okay? So you will see that the green ones now get clustered. They are given the same name, whereas here, even if they are within the same distance, they have already been assigned different names. Then I can't group them together. That's a forbidden cluster. And then I continue with this for as long as I can group something together with something which has a name. But now you see that we have 2 groups where obviously things are fairly close together, so they should probably belong to the same taxon, but I don't have a name for them. Then I apply the same principles, to cluster them up into groups. So I know this is a unit defined by the very same criteria as the ones for which we can put the name. It's an equivalent cluster, an equivalent unit, but I don't have a name for it. So I simply count it as one, but call it a pseudo species with an arbitrary number. So now I can deal with them like species. All of these species have been defined by the same principles, and this is the way to deal with the unknown diversity, to cut it into species-like units, even if we know that most of them cannot be associated with the classical name. And why this is so important is because without this, we can't deal with the unknown variation. We can't decide how many species there are in our material. We can't compare communities in terms of what species they share with each other if most of them are just unnameable and unknowable. But now we can operate with them. And what this allows us to do is, for instance, to compare species numbers across the world and to compare unknown species to previously known species by seeing how many we can name by these principles based on the existing reference databases. And one of the big results from LIFEPLAN is then that actually it is absolutely true that most species in the most diverse organism groups that we find anywhere are indeed unknown in the sense that they exist in no reference collection. They have never been collected and sequenced before. And if you somehow thought that all of that unknown diversity is in the tropics, then if you are an entomologist, you are partly right, because so here I compare for animals in the sense of the best animals, which are arthropods, insects, what fraction of species, genera, tribes, subfamilies, etc., are most likely unknown, maybe unknown, or known from before in the tropics, in the temperate zone, and at high latitudes. And we find that for fungi, it's very similar across these climatic regions. Very few species correspond to anything that we can say for sure we have seen before. Most things are either reliably unknown or most likely unknown. And even at fairly high taxonomic levels, a large proportion of the taxa are previously unknown. Whereas if you go to insects, well, at the species level, most things are unknown or likely unknown, but the proportion is still higher in the tropics than at high latitudes, which becomes more evident when you go up the taxonomic levels. So that means that if I was to walk out in Riga, where we are currently with Otso, and take a random sample of air, then it's most likely that about half of the fungal spores that I will find in there are previously unknown. About half of the fungal taxa that I will find in there are previously unknown, which is fairly impressive. Okay, I'll skip that. And this is also, this very approach is also the basis for the paper that Otso was showing, where we compare fungal communities across the world and look at how much they change with season and with latitude. we couldn't do this unless we could decide on, unless we could group all of this sequence variation into taxa and look at what differs with season or latitude, etc. And one of the results here, I mean, a basic question is, you know, do we have more species in the tropics? Well, across fungi, where most fungal species are unknown, what we can say is a resounding, it depends on what fungus you are talking about. For instance, plant pathogens, there is little seasonal variation, which is the straight red line here. The day of the year runs on the x-axis. There is more seasonality in the temperate zone, which is the green one, and most seasonality at the high latitudes, but even the peak here is lower at the high latitudes than at low latitudes. But then compared to something like ectomycorrhiza, here actually, if you look during peak season, you will have the highest species richness at mid latitudes. And you find all variations on this pattern depending on what type of fungi you are looking at. And now I fear that we will be running out of time. So I will just run through the sound patterns that Otto was briefly referring to, and say that what eLTER has contributed to by all of these audio recorders is to record all types of sounds across the year. And the main pattern here, so here are all the sites, here are all the weeks for which it has been done for all of these sites. And what we find is that in terms of the sounds produced by animals, the patterns are extremely well predictable. So if you tell me what time of the year and what time of the day you are listening to your surroundings, we can predict fairly well how much sound emitted by the living part of nature you will hear. However, if we are talking about all the sounds produced by humans and non-living nature like rain, wind, etc., then the patterns are much more noisy, and it will be much harder to say what kind of what the surrounding will sound like at this time, at this date. And also, if you are in an urban surrounding, then there will be much less silence and much more noise in terms of all types of human-generated noise. That's not very surprising if you are inside of an urban area. But what this means is that there are actually very few places on the globe that are anywhere near silent. There are very few sites on the globe where you cannot hear sounds emitted by humans. And because the rhythms in these sounds are so noisy, it means that both humans and animals are now trying to communicate against a much noisier background than before, which makes signaling much harder than before, and also creates selection pressures for modified communication against this new type of background. So if we started from this view of the objectives of eLTER, it looks like a fairly straightforward task. And what we are finding is that eLTER and the rest of us are actually faced with describing a world which is much more diverse than we thought, where most species are unknown, where handling this unknown part of nature is actually just as important or even more important than handling the known minority. And in this world, everything is also rapidly changing. The good news is that we are currently, through LIFEPLAN and through other projects, learning how to deal with this massive amount of previously unknown diversity.